Queremos continuar con toda esta conexión y poder que nos ayuden ustedes a nosotros a desarrollar todas estas investigaciones sobre diagnósticos y sobre biomoléculas que pues, el CRIP quiere escalar. Y bueno, sin más preámbulo, pues les doy la bienvenida a nombre del CRIP, a nombre de la doctora Paulina Franceschi y pues muchísimas gracias por esta presentación.
Our company was founded by three individuals. Their names are Molly, Mike, and Matt, and they were all formerly Illumina employees, believe it or not. Our headquarters are in San Diego, and we are about 5 minutes away from Illumina. And San Diego has become really, in the United States, kind of a center for biotechnology. So originally it was Boston, then it migrated to San Francisco with the University of California, San Francisco. And now we see, because of a lot of the genomics work that Illumina did, another hub of biotechnology has been in San Diego. And that's largely because a lot of the talent is there. So our founders are all from Illumina, and they left Illumina to start a reagent company. And actually our CEO, Molly, is largely responsible for the Illumina sequencing chemistry called sequencing by synthesis. So her team is really the one that developed sequencing by synthesis. And you know, when a company gets big and so large, if you're a scientist, it's hard to move your work and your objectives forward. So Molly reached a point in her career where she needed to leave Illumina to work on the projects she was most passionate about. So that's how Element was started. So she left Illumina, and like I said, it was originally to start a sequencing reagent company. And in developing the reagents, they realized their discovery was groundbreaking and it was innovative. And so they developed a more cost-effective way to sequence, about 70% less expensive than Illumina's technology. So they quickly patented that technology and they coined a new term, and the sequencing technology is called avidite-based chemistry, ABC, which is different than SBS. And really the avidite-based chemistry gives us a lot of scientific advantages in terms of accuracy, but it also lowers the cost. So with this, they did a lot of research and development, and about 5 years' time, we brought our first instrument to market. So from 2017 to 2022, it was in development, and then we placed our first instruments in 2022. And international appetite for our technology has been more than we expected. So already in just 2 years, we have over 500 instruments globally. So we have instruments in Asia, Europe, obviously the United States. We have instruments in Australia, Africa, Malaysia, Singapore, Thailand, Turkey, all over. And we entered Latin America intentionally last. And that is because we are a relatively small company. We have 400 employees. And so we don't like to enter markets if we cannot support the market. So when we mean support, we like to do a lot of outreach and education, like having me and Solomon here. And we weren't quite ready for that in 2022, 2023, and the majority of 2024. But we're ready now, and we're ready to support you, and we have great partners like Rochem to do that. So I will also say, we started as a sequencing company, and from day one, our leadership said we are not competing with Illumina. We are not a sequencing company. We are a biology company. And you will see the innovation we put into our instrument is that, yes, it does NGS, but it has transpired or transformed to be a multi-omics instrument. So we do now, in the same instrument, in the same box, we can do NGS for all of your sequencing needs, but it also does multi-omics. So we can do RNA, we can do protein, we can do morphology in the same instrument. So it's, the analogy I like to use is sequencing is like an old telephone where you pick up and it's attached to the wall. This is a sequencer. We have really developed a smartphone. So our sequencer, you can think about it as like a smartphone. It does email, it does pictures, it does video, it does applications, it does social media. This is how I want you to think of AVITI. It's a multi-useful box. Some of the unique advantages of the instrument is that it has two sides. So you can do sequencing on side A, and you can do multi-omics on side B. You can do sequencing side A and sequencing side B. Some of you might be familiar with Illumina's instruments. Our instrument is a benchtop platform, a desktop platform. So it would be most comparable to like a NextSeq in terms of throughput and volume. And we like to say it's two NextSeqs in one. So it has two sides, and the capacity is similar to two NextSeqs. And it's super flexible because you can, like I said, you can start a run on one side and then start a run on the other side a couple hours later. Here's kind of the timeline. It's a very fast pathway to commercialization. We were founded in 2017, and we placed our first instrument in March of 2022. So in that time, we have continued to develop and innovate, and really the culture of our company is very research and development. We are a company built by scientists for scientists. And I have to be very transparent, that sometimes is a challenge because the R&D team is really the culture and the fabric of our company, and they want to continue pushing the boundaries and making new innovations. And the commercial team says, wait, we're good enough, let's go sell the instrument and make money. So we have a very good combination of innovation, research and development, but also being able to support a finished product. So you could see transformation of the company, and just last year is when we announced the AVITI 24, and that's the same instrument as our original instrument with a lot of enhancements. So this is basically what we've done to NGS. I mentioned we have over 500 instruments placed all over the world. The foundation of the instrument is that it's affordable. We've been able to achieve a very effective cost for sequencing because in a small benchtop desktop platform, we can achieve the same cost as the big factory scale NovaSeq instruments. So if you think traditionally about NGS, the only way to lower the cost is to have more samples. And that is because of the chemistry. The chemistry is inherently expensive because of the SBS. Dr. Solomon will get into some of the details on how our chemistry, we have over 500 patents either applied for or granted, and it's largely due to the innovation in this instrument. It's flexible, I said it's got two sides, and the quality is unmatched. We now have gone from Q30 accuracy to having Q50. So it's very reliable and accurate, and I think you'll enjoy that. So we have 3 iterations of the instrument depending on where your laboratory is. We have a low throughput instrument that does up to 500 million reads per side. So if you ran side A and side B together, you get 500 million times 2. And we have the standard AVITI, which is 1 billion reads per side, so 2 billion total. And then we have AVITI 24 that does sequencing 1.5 billion on each side, so 3 billion total if you do sequencing, but it also does multi-omics. And I will leave you with this, is that we designed the platform, as I said, it was built by scientists for scientists, where you can upgrade. So if your laboratory wants to start with an LT, and 6 months later or a year later, you can do an upgrade to the standard AVITI. And it's a field upgrade, you don't need a new instrument. And the same with the standard AVITI, you can go to an AVITI 24. And we send a kit, we send an engineer, and we upgrade the instrument in your laboratory. So Dr. Solomon has many more slides. He's much more knowledgeable on the applications that you guys will be using. So I'd like to introduce Dr. Solomon.
So a little background about myself. So I did my PhD and postdoc at MD Anderson Cancer Center. Has been working in research almost 10 years or so before transitioning into multiple biotech companies. I used to be a sequencing specialist for Illumina before joining Element. So a lot of background in microbiology, standardization, all of those. So if you have any questions regarding any of the applications, please feel free to ask those. But in terms of the innovation, when we started our first platform in 2017, it was actually designed not for sequencing. It is with the intention of doing multi-omics and spatial, and we will highlight each of those. But the main innovations that, as you see in the first panel, is the surface chemistry. Not only we can attach RNA, DNA molecules to our flow cells, but we can attach cell, and we will be attaching tissue in early next year as well. So that is one innovation. The second innovation is, let's say you're doing exomes or targeted sequencing. Right now, you do a lot of bead washes, multiple manual workflow. So we innovated to actually have that on the instrument itself. And I'll highlight that in the middle of the presentation. We call it the Trinity workflow. So not only it reduces your time by 1 day, but it also have a lot of advantage in your SNP and indel detection. So we'll highlight that. The chemistry for our sequencing is another innovation that we brought in. So typically for Illumina, you're using XLEAP or standard SPS chemistry, and we will highlight why this chemistry is much better and why you should care for your research. And I'll highlight with some examples. And then of course the field of view, the imager, all of that is innovated, including the software, and particularly the software allows you to be able to adapt multi-omics as well as if you have low diversity sample, let's say you sequence a specific genomic region for a certain gene of interest, it is low diversity. So our softwares are able to adapt to that as well. And all of those benefits, you find it on our sequencer as well as our multi-omic instrument. So I'll highlight all of that as we go. But first, let's focus a little bit on the sequencing side, and then we're going to transition into multi-omics on the second half of this presentation. So as Vasili mentioned, our sequencers has dual side. Side A and side B are completely independent. The fluidics is completely independent. So the only thing they share is the camera. So the camera is automatically adjusted by the software. So you don't even need to worry about that. So let's say you're running side A, and then another sample come in, you could run side B at another time. Or if you want to start two sides at the same time, you can do so at the same time as well. So it gives you flexibility of how you want to process your sample, right? You can use different flow cell configuration, different cycles, it doesn't matter. So it is completely independent sites. But what makes this sequencer more important for you is our quality. One, it is industry leading quality, greater than 90%, but most of our reads are in Q40, which means the error rate is 1 in 10,000 rate, and I'll show you that with data. Our duplication rate is one of the lowest. You compare MGI, you compare Illumina, it doesn't matter. We offer the lowest duplication rate. That means you get more real estate for your sample that you care about. And of course, compatibility with low diversity sample. I know you work with microbiome, for example, you want to do 16S sequencing. This is very targeted region. So what you do is to increase diversity, you add 5x. And that you're not going to use for your data. You throw it out. This is just for imaging purpose. You don't need to spike a lot of 5x with our instrument. So it increases your effective coverage. So for your microbiome applications, that makes it more robust. Or if you're doing, let's say, methylation sequencing or other things for oncology, that becomes a benefit as well. But we have 3 different flow cell configuration. We have the low, the medium, and the high. Let's say you're starting with a small microbiome project, you can use the low flow cells. And if you want to expand your project, you can use the medium and the high flow cells depending on your throughput. So we have different cycles within each of those flow cells. You could use 2x75, 2x150. It doesn't matter. You could use paired end, single end, asymmetrical end. You have that all possibilities. But also, each of this flow cell comes with individually addressable lanes. That means you can put 2 projects on the same flow cell and run them at the same cycle. Or if you want, you could use the same barcode and then pull them into separate wells and use the same barcode on different lanes. So they're completely independent, giving you more flexibility on sample processing. But for microbiome or viral sequencing or other applications, certain times you may care about longer reads. So 2x300 cycle kits are there. So let's say you want to sequence HLA regions or immune repertoire regions or do microbiome sequencing. All of that can be accommodated with a 2x300 cycle kit, and they come at 100 million and a 300 million range, and completely very, very cost effective compared to the MiSeq. Any of the MiSeqs, the old, the new, this is very cost effective compared to those as well. So with all this flexibility, then what can you do with the sequencing instrument? So you could do whole genome sequence, you could do liquid biopsy assays, you could be able to do single-cell sequencing, spatial transcriptomics, bulk RNA sequencing. You could do exomes, targeted sequencing. You could do epigenetic assays like methylation sequencing, ChIP-seq, ATAC-seq, cut and run. You could do microbiome viral sequencing. You could do immune repertoire sequencing. The list keeps going on. So the bottom line is any sequencing need you have fits within this instrument. So basically it is catered to your needs. All right, so the question then is, let's say you're currently running NGS application. You're doing oncology research, microbiome research, but you don't want to change your workflow. So how does this instrument adapt to your needs? So we have 2 different workflows here. So one, if you have your own library prep that uses Illumina style library, that means P7 and P5 adapters, those are third-party libraries, can go directly without any conversion into our instrument. So you don't have to change your library prep workflow. If you want to use our indexes and adapters, you also have that option. Why our indexes are needed, for example. You don't need to change your workflow, but let's say you're doing a very specific region sequencing, and that's an amplicon sequencing. And you want to understand what is the diversity, what is the mutation there. So because it is a targeted region, it is low diversity. Our adapters and indexes have random 3 nucleotides that are integrated. So they naturally will create diversity for you. You don't need to spike in 5x. But if you want to proceed with your existing workflow and you don't want to change it, you have a third-party compatibility with our platform. So you could just simply load your library onto our instrument. What is changing is the sequencing chemistry, which I will highlight. And then once you're done with the sequencing, you get the basis file. And we have a free software to convert it into FASTQ. The FASTQ goes into your pipeline with no change. So no change on your library prep, no change on your bioinformatic workflow. The only thing we're talking about is in between. And that increases your quality, lowers your cost, and then gives you flexibility. And I think it's a no-brainer why you should care here. But in terms of the sequencing workflow, this is our flow cell, 2 lanes. And on the surface of this flow cell, you have oligos to capture your library. You float in your library. This is done automatically on the instrument. You amplify that original molecule 1,000 times. And I'm going to pause it right here quickly. So let me explain why this is important compared to the other sequencing part. So notice this is like amplifying your original molecule 1,000 times. And we call this a polyny. So in this step, there's no PCR. So if you're comparing it with SBS or XLEAP chemistry, you have a library, you PCR amplify, create a copy, those copies get PCR amplified, you create another copy to create the cluster for SBS or XLEAP chemistry. If you're doing that, let's assume you have an error in the first copy. That means you keep amplifying that error again and again, so your cluster is mixed color right now, and that cluster is wasted because it has a lot of error. So on our technology, rolling circle amplification, PCR-based errors are completely avoided. Because of this, you get up to 10 times more quality with our workflow. So with this, you're amplifying the original molecule. If an error happens in the first copy, you don't find it in copy 2 or copy 3 because it keeps amplifying the original molecule. So because of this rolling circle amplification, no PCR error. So that's our first advantage. And this is similar to MGI. They use also rolling circle amplification. No difference here in that case. But the sequencing chemistry here, which uses the avidite-based chemistry, is another difference. So we use avidites. These avidites, as you can see, have multi-valency. Multiple valency here, the same basis. So let's say this is A, this is A, this is A, and A, right? So our avidite has like tentacles, so each connected to the same base. Let's say 4 bases of the same kind. Why that is important compared to SBS? So in SBS or exit chemistry, you have one nucleotide with one fluorophore, and one polymerase binds to that. So the binding is so poor, you have to add more and more reaction to push the reaction to keep it stable. So that means your XBS or XLIP chemistry costs a lot more than it is needed. With this has multi-valency, right? One avidite binds to different copies, at least 4 in this case, or we can make it 6, we can make it 8, we continue to work on it, but 4 is what we use for sequencing currently. So with 4 valency, that means one avidite is stabilized by 4 polymerase. So that means the stability is once it binds, it doesn't fall off. So you don't need a lot of reaction or a lot of expensive reagent to push the reaction forward. That means because of this innovation, you lowered your cost of sequencing by 50 to 60%. So we're not giving you a discount to compete. We actually innovated, so you benefit from that innovation. So this is the second differentiator from all other sequencers. And in terms of like how the sequencing or detection then happens, so you float in the avidites bind to multiple copy at the same time, and then we detect the signal. And this happens for millions of colonies across the sequencing flow cell. And that's how we get our bases, and then we convert them into FASTQ. So completely different from SPS, where we use rolling circle and avidite-based chemistry to give you the highest quality. So the benefit of that is then you can see a lower duplication rate. All these 4 runs are AVITI runs. So run after run, you can see the duplication is below 1%. But for MGI, that is at least 2 to 3 times higher because they're using rolling circle amplification, similar kind of optical duplicates, but we're like twice lower. With Illumina, you can see that it is at least 8 times, in some applications to 20 times lower in duplication. So the lowest duplication you get is from our platform. And of course, index hopping is almost negligible. So this means, let's say you are a sequence read from one sample, is assigned to another sample it doesn't belong. That's what we call index hopping. So that is higher, around 4% to Illumina platforms, and ours is almost negligible because of rolling circle amplification. So both of these will help you to increase your effective coverage. You can add more sample, lower cost, or you can have more coverage per sample. So both gives you that advantage. But it is one thing we say, it's another thing when a customer actually sees it in their own data. So this is a customer from New York. They were using this RNA-seq for single cell, and then they have like a very similar basis, so it is low diversity. So because of that low diversity, they have to have about 25% of phi-X for their assay. When they converted to AVITI, they only needed to use 2 to 5%. The 20% that they were wasting now becomes useful for their reads. Then, if you compare the usable reads after your sequencing, after your alignment, after you filter duplication, after you removed errors, 65% of the data is usable. For us, 90% of the data is usable. Then, if you compare it into a flow cell perspective, the median flow cell they use from AVITI or Element gives them 450 million out of a 500 after removing duplicates or so. And then the next seq, the P2 flow cell supposed to give them 400, gives them 260 million usable data. Now let's put it into cost perspective. You pay X amount for Element, 3 times more for the newer chemistry on Illumina, 4 times more for the older chemistry, SPS. So think about it. You're paying 3 times more to get twice lower the needed through the usable data, and then 10 times lower the quality. Which one would you have preferred? It's a no-brainer. Why customers keep switching from using old platforms to a newer AVITI platform in chemistry. So you could also see that the chemistry is comparing all platforms. MGI and Illumina are in the Q30s, which means 1 in 1,000 error rate. Our standard sequencing is in Q40, which means 1 in 10,000 error rates, which is 10 times higher quality. But we also have the first commercially available Q50 kit that you can use for rare germline mutations with no PCR whole genome sequence, or if you're doing low-pass whole genome sequence, that can be used for that. So if you have very, very ultra rare allele variants that you want to detect, you could use that kit. But for most other applications, the standard sequencing kits are more than sufficient. So, and they're already 10 times higher in terms of quality for your assays. Yeah. So in terms of the sequencing step, what happened is the first initial, once you press go on the sequencer, there's an amplification step which takes about 4 minutes. It amplifies the library. And then once the library is copied 1,000 times, then you float in your sequencing reagents, right? So the binding first happens. So you float in the avidites, they bind to the correct base. We do it base after base. So once they bind, we detect, as I shown you earlier, and then that binding is done with a separate polymerase. It doesn't extend, it just binds. And then once we detect the signal, we remove the polymerase, we remove the avidite, and then we put a separate polymerase with a nucleotide, which is blocked at the 3' end. So that steps the sequence by 1 base pair. And then once we finish that step, we remove the blocker, and then the cycle continues again, back and forth, back and forth. And if you want to see that, I'll show you the slides at the end. Yeah. So because of all these efficiencies, then you benefit from cost and quality. But I want to put some examples of applications that we have used our platform for. And one of them is the oncology application. So you can do somatic mutation from solid tissue. If you want to do heme or blood cancer detection, you could use it. For cell-free DNA-based early detection, you could use it. And now UMI-based application for ultra-rare variants, and then as well as MRD, the platform is being more and more being used. So I'll show you some examples of that data. So this is data from Burning Rock. They have this oncology panel that targets 520 genes. So they already have NovaSeq data set, and they want to compare that with our Element platform and see if there's any difference between those platforms or any advantage. So when they compared at different targets, the allele frequency, you can see the correlation is basically similar between the two platforms. When they compare copy number variation, for each of their targets, the copy number is no different. So they get copy number variation similar. When they looked at tumor mutation burden or MSI, microsatellite instability, you can see the performance is exactly identical with that of the NovaSeq. But when they look at effective coverage, you can see that the effective coverage is more higher to Element versus the NovaSeq itself. So that means our platform is very sensitive and can actually go into lower allele frequency than the NovaSeq itself. So for oncology applications, you could use this platform at a benchtop with no need to batch more samples. So for the NovaSeq, you have to wait until you fill the flow cells. For us, it is a mid-throughput instrument. As samples come in, you increase your turnaround time for those kind of applications. But we also took a look at comparing the NextSeq with our platform for hematological samples. So you can see that the allele frequency for the NextSeq 500, which is typically used in this case, is at this allele frequency, which is about 1–2%. And when they compared it with our platform, the allele frequency goes to about 0.1%. So on a benchtop instrument, you could go as low. And when they compared the error rate for each base transition, our error rate is lower, and then we have higher coverage at each target that they compared. So for any oncology application, this platform outperforms or is similar to existing solutions. But when you want to look at a very targeted region and you have to have UMIs for those applications, you need more coverage. So let's say you want to go to 1% or 0.5% allele frequency. At each coverage, we compare the Illumina platform to our platform. you could see that you have better recall for our platform compared to the alternate platform. So oncology application is already shown this platform is better. But you want to do microbiome sequencing. So this is our whole genome sequence for microbiome. So you can see our quality is above Q40, which is 10 times higher quality than alternate platform. So for any microbiome sequencing, you can use this. If you want to do viral sequencing, you could do viral sequencing and detect it. Our correlation between our Illumina as well as the AVITI is 0.99. So there's no difference in terms of that detection. And we can detect infections that are existing. Let's say if it is COVID and then you have additional infection, you can also pick that. So we used it for viral sequencing. We used it for microbiome sequencing. So the sequencer is compatible with any application that you guys might be interested currently. So with that, that's the first section. And I want to talk more about the innovation that we're different rather than just the standard comparison of sequencing. So what makes us different from all other platforms is the innovation that we bring in. So in terms of Trinity, this is a workflow for hybrid capture assays, exomes as well as targeted sequencing. So currently, you make a DNA library, whole genome library, and then you have a target probes that you get from Twist or IDT, and then you hybridize overnight, 16 hours, and then you do continuous bead washes to clean it up, and then you may lose a bead or something during cleaning, and that means more error from run to run, and then you do post-hybridization PCR. If you want to follow this workflow, fine. Both our platforms, whether the AVITI or the AVITI 24, can do the sequencing. You don't have to change anything, you can do that. But why do you care about the Trinity workflow? So once you do the DNA library and the probes from Twist or IDT, you hybridize for 1 hour, already to saving you a day of workflow. And you don't need to do any of these. Everything after hybridization, you load it onto the sequencer, press go, and walk away. And you can do other things. So it is workflow innovation. However, when we look at the data, we compare Trinity, Freestyle, and InSolution, the standard workflow in solution, as well as on the flow cell workflow. So you can see that the main difference I see is the indel ratio. So if you want to detect indels, then it is much better to use the Trinity workflow because your indel detection is much higher than the traditional workflow, insertions and deletion. There is a slight advantage for SNP calling as well. So this is single nucleotide polymorphism. So if you are interested in insertions and deletions, the Trinity workflow would be much better, but you can also use it just to reduce your turnaround time for applications that require that. But this highlights a very good, clean example where we mix the sample to genomic data sets in a known ratio. We know exactly what to expect in terms of insertions and deletion. When we did the traditional compared to the Trinity workflow, you can see that this insertion completely is not there on the solution part. When we did it on the Trinity workflow, on the flow cell, we recovered that insertion. In this part, there's no even mapping to the deletion region. So not only it facilitates your turnaround time, but it also gives you advantage for insertion and deletion to use that. So if you're doing targeted sequencing, that would be my recommendation. But the most exciting part is the AVITI 24. Sequencing, everybody does sequencing. Trinity, we do it. But AVITI 24, nobody else doing it. So it is only an element solution. And the reason why this is exciting is it would have cut my PhD by half. Because to do this standard sequencing, I used to do whole genome sequence, RNA sequencing, epigenetic sequencing, all of that. Then when you combine it, the correlation is very poor. And you know it, everybody knows it. Because the data comes from separate samples. Even though individually they are great, when you combine it for multi-omics, the correlation usually do not relate. So there's a lot of layers we don't understand. With AVITI 24, you get everything with one experiment. So I'll show you that. So the whole premise of AVITI 24 is you combine morphology data with RNA, protein, phosphoprotein information, all from the same cell at the same time. You don't need any library prep, and it is done in 24 hours. So there's no other platform that does all of these in one go. So it is cost effective, but it is the holy grail, where there's no library prep, Just grow your cells, fix them, and proceed. And then you get your data 24 hours later. How do we do that? So before I go there, most of you may be doing single cell, maybe doing spatial transcriptomics, maybe doing protein detection for Olink or DULISA. All of them generate libraries. So if you want to do that, that's fine. You can sequence on AVITI24 because it's a sequencer too, right? But the data you get is segregated, so you have to piece it together bioinformatically. So that means more time for your bioinformaticians to be able to sort things and then identify correlation. So you don't have to do all of these because the data now with AVITI24 will come combined, so it saves you a lot of time and the data is more correlated as well. But if you want to do that, the AVITI24 increases the throughput by 50%. The same flow cell on a standard sequencer that gives you 1 billion, if you use it on AVITI24, it gives you 1.5 billion, and you don't have to pay more for it. That means you're saving additional sequencing costs by 30% for your application. So even if I'm doing sequencing, I would use AVITI24 because it reduces my cost by 30%. Why not? And there's no penalty in quality, there's no penalty in performance. So if you want to do all the assays the way you are doing, you can use the AVITI24 for cost as well as quality. But the power of this platform comes in not from the sequencing side only, but you could use dual side to get multi-omic data set, right? So you're getting RNA, morphology, protein, phosphoprotein in a spatial context. You could do time course, you could do dosage-dependent assays in one go. And there's no need for library prep as well. So you could also sequence directly from those cells. So there's no other platform like that, so I'll walk you through each of those steps. The workflow is very simple. You have 3 different flow cells. So this one has a 12-well flow cell. That means 12 samples can be accommodated at the same time. This one is a 1 million cell, can be done on one flow cell. And that 48 well accommodates 48 different conditions. Each well have 10,000 cells, and the 12 well, each well has 50,000 cells. So now you grow your cells on those flow cells, put them on an incubator. Once they reach 50 to 70% confluency, fix them, and then assemble this. After the cells grow, fixing and assembling takes only 45 minutes hands-on time. This is the hands-on time you're talking once the cells are ready. So you fix them, you assemble, load it onto the platform, press go, and walk away. And then 24 hours later, you get multi-omic information for each of those cells in each of those wells for each of your sample. So an example is shown here. So this is a 12-well flow cell. On each well, as I mentioned, 50,000 cells can be accommodated. So in this design, you can use it as a time course. So you can see that the wells for different samples are organized by time. And then you can also treat drugs, drug A, drug B, at different time points. But the wells can be used for different stages of differentiation, or they can be used for the same sample, different reproducibility as well. And for each of those, you get those modalities and in a spatial context. And I'll highlight the different ways how you can benefit. And we recently enabled directly sequencing from those cells the RNA as well. So you could have, let's say, a mutation hotspot, you want to sequence it, you design one probe, and then sequence 100 base pair from those same cells while you're getting the other modalities as well. So that feature is now enabled. So there are 2 kits for this platform to be used. The first one is the Teton kit, which is a fixed panel. So the fixed panels will give you all this, I'll highlight, and then the second one is the direct in-sample sequencing. So let's walk you through the fixed panel. So the fixed panels are with the Teton panels. So the fixed panel have 6 morphological features that you get. Cell membrane, nucleus, and actin, we use it for cell segmentation. But out of these 6 morphological features, we extract more than 600 different characteristics. So that means it gives you cell paint kind of assay. So if you're doing drug screening, development, maybe the impact of that drug, how does it change morphology? This will pick it up. So those 6 morphology, they can give you over 600 features that you can compare. And then the second is the RNA. Each of those panels that we're talking about, cell cycle, apoptosis, immuno-oncology, and neuroscience panels, each of them will have 350 targets. And the target lists are available on our website, and we're happy to share those. And you can quantify the using the barcode sequencing up to 350 RNAs within that pathway. For the protein detection, each of the panel comes with 50 different targets. It could be cell surface, intracellular, or phosphorylated states. So each panel has all the 3 components that are available. But then the panels are listed here. What if like you're doing immuno-oncology or neuroscience and your target is missing? What do? So you have a customization capability. So you can decide which protein you want to look at, and you can do that up to 88 plex of your own choice and add it as a custom on top of the 50. So now you're looking at 138 proteins for each of those cells for each of the pathway you want to look at. So customization is possible. You also have 9 areas, focus panels. Each has 24 plex protein. You can combine 3, and then you can use one of those and then customize it. You can use 2. One of them could be your own custom 24 and then mix it too. So there's a lot of customization that you could do if you're not finding your target on those fixed panels. But I know some of you do neuroscience and immuno-oncology. Those panels can be easily used and there's no additional step. Just plug those, the cartridge will come, you grow your cells, fix them, and then load them, press go, walk away. So 45 minutes hands-on time on your side. So that are the fixed panels. But now we are enabling direct sequencing from those same cells as well. That is what we call the Teton Atlas kits. The low output version is already available. Next month, we are enabling the high output, which gives you 3' whole transcriptome sequencing. So think about it, Chromium, where it does 3' gene expression, now we do it with actual sequencing on the platform. It's not probe-based. So direct sequencing from the cells are possible. So as you can see here, we're sequencing from the cells. We can detect the bases up to 100 base pair. You can combine it with the morphology. You can combine it with protein information for each of those cells at the same time. So this is what we call direct in-sample sequencing capability. Yeah, so what we are able to do, how we are able to do the direct in-sample sequencing. So you design a probe, which is already designed for you, that sits at the 3' end, and it sequences 100 bases, or in this case, 50 base pair for the 3', and then we circularize it, and then we use rolling circle amplification, the same thing that I mentioned for the sequencing, use AVITI for sequencing. So our 3' gene expression is not probe-based. So that means if you detect a signal and the probe shows you positive, it may not be necessarily a true target because it may be randomly sitting somewhere and you detect. With our case, you're actually sequencing. So you take this 50 base pair and align it to a reference. So anything non-specific will be filtered out. So this is a better comparison in terms of 3' whole transcriptome RNA information from that same cell. And now that you have that, let's say you want to target a specific region. Let's say a mutation hotspot, splice junction, fusions, or so. You could design one probe, sequence 100 base pair, do rolling circle amplification, and use AVITI to detect that mutation as well. So in any combination you want to use the sequencing part from the cells, you can combine it with morphology and protein data set at the same time as well. So here are some examples. So this is CRISPR-based perturbation. So where we're detecting which cells are expressing which guide. So we're sequencing 20 base pairs of the guide, and you can see different cells are expressing different guides. Not only we know that, we can then combine that in this cell, what is changing versus the other cell in terms of protein and morphology. You get that for each of those cells at the same time. If you want to look at, let's say, splice junction or mutation hotspot, design a probe right here, sequence 100 base pair, and identify that. This is also done directly in the cells. If you want to do whole transcriptome, you use our whole transcriptome kit that will be coming out next month for early access, and by New Year it should be ready commercially. So you can then align that 50 base pair to a reference and do the quantification of gene expression. So in any combination you want to use this, that feature is available, and again, no one else is able to do that currently except Element. So if you have, let's say, multiple cell types, you can grow them together, culture them, and then you can easily separate them using that whole transcriptome application. So we have a design tool freely available for your RNA targeting or for protein customization. So you can take advantage of that. And then once you get your probes from Twist or IDT for the sequencing part, just add them here. If you're customizing protein, it is your own antibody choice to customize. If you don't want to use the custom only, plug that in here, load it, grow your cells, fix them, and then press go. Depending on whether you want to use fixed panel, 24 hours. If you're going to use fixed panels plus the direct in sample whole transcriptome and all that, that is about 72 to 60 hours. And then you analyze the data. And the data is compatible with already existing single cell pipelines. So there's no issue in terms of how you want to analyze it downstream. But we also have softwares to help you with visualization for the data as well. So currently what we are able to do is adherent cells, cell suspensions, and PBMCs are compatible. And we have 4 different panels, and we're going to add more and more. And then 3 different flow cells for your experimental design. You can use them in any combination you want. And then we have direct in-sample sequencing capability, which no one else is offering. You want to do CRISPR screening, you want to do immune repertoire sequencing in combination with microbiome. Think about one customer actually is currently trying to do co-infection, immune cells infected with virus or bacteria, and then which cells are responding to that. You can sequence the T cell receptor from that and then identify which microbiome is there using the protein marker, and you can be able to address that question immediately. No one else has that capability with such platform. So for different applications like that, you can think of it, or for immuno-oncology or other disease applications, this is also available. And then you can customize it the way you want, and we're more than happy to help you. So there are different approaches you can follow, right? So the platform first is a sequencer, so you can do any of your sequencing to get bulk information. But let's say you want to use the whole transcriptome 3' gene expression. In your cell types and your treatment, you look at the whole transcript, which is unbiased, right? So it will tell you what expression differences you're picking up. So then once you identify those, you could use the fixed panel to target them into that particular pathway and then get a more refined data set, if you will. So let's say you're collecting different samples from different regions. You have 12 wells. So you put the cells in the 12 wells, treat them with the same treatment that you want to do. It could be treatment with different drugs or not, and see what expression change for each of those are happening because those are separate samples in each well, and then you have thousands of cells to compare to, right? So that gives you an unbiased whole transcriptome information plus morphology plus protein, right? So then once you see the expression change, which is unbiased, then you know which targets to look for specifically. Then you can get antibodies for those particular targets and do a second experiment. Now this time it is more focused to those targets. So you get the antibodies, and then we have the coordinating molecule. You do affinity binding for 2 hours, and then those targets for protein are now available. Then you could also use the targeted feature where you want to look at a specific area to sequence. And hopefully, if you're doing, let's say, RNA-seq or bulk whole genome sequence or other thing previously on that same platform, you have information where mutation or diversity is happening. So you could take design a probe and then look at that particular area with this inside the cell sequencing capability. Because you can customize, or you could just use a whole transcriptome approach, right? So it could be 1 or 2 steps before you narrow it down to a specific target for that, if you don't know anything. But if you know exactly the target, then you get the antibodies and then put them in. That will highlight the protein change and the morphology change in the treatment within that. So this may give you an idea a little bit how we did this. So this is like what is happening in non-small cell lung cancer patients, and then 2 different drugs are used. So generation 1 and generation 2. But we don't know why generation 1 is weaker than generation 3. So we wanted to understand that. So we used a panel. So this is the design. 2 drugs, one for 1 drug, the other for 2 drugs, and we did time course. And then we fixed them at different time. And then we treat them with EGF to activate those cells. And then once we fix them, we assemble the flow cell. And we used an apoptotic marker to be able to see if cells are dying or not. So those are already a fixed panel that is existing. And this is like a drug for oncology, so we're looking at cell death or cell survival. So we already have the panels that we could use. If you don't, that's where you get from the total transcriptome to narrow it down to what you need to look for. But in this case, we know we're looking for apoptosis marker. And then we did the assay. So we looked at cell paint, morphology, protein, and RNA for each of those across different time points. And then here's the data together. So based on those, different cells are responding differently based on their color. Based on time of response, they're also responding differently. So when we looked at overall bulk, you can see that at different times, the cells are dying more on generation 3 versus generation 1. So you can see more cell days happening at the 24-hour time period. It is going to come right now. You can see more are dying versus that one. So we identify, okay, cells are dying here, not dying on that one. But then we looked at combining all the protein morphology data and identify in each of those pathways what is changing actually. So you can map it because you're getting all that information from the same cell at the same time. So when we mapped all our data, what we notice is cyclin D overexpression here in generation 1, while cyclin D is not overexpressed in this one. So we said, maybe this is cyclin D dependent overexpression. So then we said, okay, if it is linked with this, why don't we use an inhibitor for this one? Because cyclin D together with CDK4 phosphorylates RB, RB then releases E2F, and this leads to cell survival. But if we block this together with this, can we rescue the phenotype? So just by looking at our multi-omic data, we identified that, right? So then we did a second experiment where we combined the drugs with a CDK4 inhibitor. And then we loaded the flow cell, looked at the E2F1 expression and the phospho-RB, because now we know exactly what to look for. So we looked at those expression, and we can see that expression is dropping. So you can see here, with no combination, the normal control, with only the first generation added, and then with the combination drug, you can see more cells are dying, and we're rescuing that. And the same study was published, which took months of work to get to that answer. We just did it with 1% 2 flow cells in 1 week. And exactly, the cost saving is significant as well. So the same phenotype is, this is a published data, and we did it separately just in 1 week. So you can narrow it down, you can start bigger with whole transcriptome, and then you can narrow it down to specific targets you look at, because you have a customization capability with your assay. But remember, it's a sequencer too. You could do whole genome sequence, whole transcriptome, look at overall profile across your sample, and then once you identify a certain pathway that is impacted, then you come to the cellular base and use the 12-well or the 48-well for, I'm not sure, there are more than 48 states or countries in Latin America or Central America, right? So one flow cell can take all the samples from different regions, and you could be able to treat that in the same condition and see what is changing in those at a particular panel, or when we have the whole transcriptome. That would be my first suggestion, is use the whole transcriptome. And then based on that, you can also look at morphology changes and identify which samples are behaving differently. And then you look more refined customization. And we can help you that specifically if you want. And they're also here to support you as well. So finally, tissue. I want to finish with tissue. So the same platform not only does sequencing, not only does single-cell multi-omics, not only does direct sequencing from the cells, but in 2026, mid-2026, we're going to do tissue on those as well. So fresh frozen FFPE, and if you have existing mounted tissue on a glass slide, that's also compatible. So again, you have 20 centimeter usable area on both sides of the instrument. Just to give you an idea, one flow cell is 10 centimeter square usable area. That is 5 times more than the Xenium from 10X, which is used mostly for tissue-based application. So you have more advantage in cost saving and multiplexing different tissue types. Here is some of the example tissues that we have analyzed. You can see small intestine, brain, lung, breast, colon. Different tissue types has been tested so far, and we continue to test it, and this will be ready by at least half of 2026. It has 3' whole transcriptome that we talked about for single cell. We're going to have it on tissue. We're going to have target capability. Let's say pathologists probably look at 10 or 12 different targets for different samples. They don't need a lot, even though the platform has that capability. If they want to customize what protein to look for in those tissue, they can customize it. And they also get 3' whole transcriptome data. They also get morphology information and all that. So if, let's say, they're looking at fresh frozen colon sample, so you could get morphology information, identify that, and then you can overlay that with the whole transcriptome information. And if you want to look at, let's say, healthy versus cancer cell and compare the expression, you can do the expression on those tissues and quantify the differences. If you want to do, let's say, protein detection, so this is a FFPE sample, and this is not immunofluorescent, by the way. This is avidite-based sequencing chemistry, which is much better than immunofluorescent because there's no background. You can have multiple targets at the same time. You can scale the signal up and down because we're using our sequencing chemistry, not immunofluorescent. And you can see beautiful morphology, structure, and proteins that are there. We also will be doing tissue segmentation at single cell level. That is being worked on. And finally, if you want to do FFP, if you want to do fresh frozen, if you want to do organoids, you could do it on the AVITI 24. So almost everything, tissue, single cell, and sequencing are possible. So finally, I know it's just been so long, but it is the end. So you could do next generation sequencing from whole genome, exomes, targeted sequencing, metagenome, viral sequencing on one side or the other side, or you could do multi-omics for single cell, eventually for tissue on one side or the other side, or you could do, let's say you want to discover, you could run whole genome and RNA on this side, and then while you're doing that, you could do cell-based targeted sequencing on the other side and get the answer at the same time. So hopefully this platform gives you an idea of what is capable and what is different, and hopefully you see the innovation that Element is bringing. Yeah. So yeah, there's multiple applications that are going on. So some are using, for example, iPSC-derived differentiation. So they do differentiation studies. Others are using it for toxicology application. So which toxins are more beneficial or resistant or not. So they do a lot of toxicology for environmental studies and all that. There's a huge interest in microbiome host interaction. In fact, when I gave a talk at Nebraska University, they actually bought AVITI 24 after that. So their interest is there's a specific strain of Staphylococcus that no one knows which T cells respond to that. So no one knows that, and they couldn't figure it out. So the only way they can use is using AVITI 24, because if the host is interacting with that specific strain, you can pick it up with AVITI 24 and sequence the T cell receptor so that you already would know what to design to block that specific strain of Staphylococcus. So for that, this is the only platform that they were able to use, and immediately afterward, the university ordered this one. So there are different applications like that. Biopharma use CRISPR-based perturbation. So they knock out specific target proteins and look at what drugs would be useful or what is changing, and then test their drug combinations as well. So Eli Lilly, for example, running that kind of assay right now. And so different application in different ways. Toxicology, drug development, compound screening, microbiome, co-host interactions, all of that, or different for research, different stages of differentiation, reproducibility, correlation between proteins and RNA in certain specific conditions. So you name it. It is very broad. There's no one particular niche, oh, this is only for this. It could be applicable for any different kind of research. So if you have a project, don't be shy, let us know. So we'll see how this platform will be able to help address those questions that you may have. All right, so we're gonna be here. If you have any question or if you wanna look more data, we'll be happy to do that. Thank you.
Mucho gusto, yo soy Nicolás, licenciado en biotecnología y cursante para una maestría en biología molecular por la Universidad de Panamá. Les voy a presentar sobre nuestras soluciones integrales para el campo de genética, pero verdaderamente es para todo lo que sería biología molecular. Nosotros como Rochem Biocare Panamá somos un proveedor líder en el campo de biología molecular y también todo lo que esté relacionado al área clínica. Tenemos ya más de 35 años en el mercado panameño y somos una filial de la compañía Rochem Group, fundada en Suiza. Nuestra misión siempre ha sido proveer información oportuna, confiable a nuestros clientes a través de un excelente servicio al cliente de nuestros productos y siempre generar una buena calidad para la comunidad, el investigador y el grupo en general. Nuestra visión siempre ha sido mejorar la atención al cliente, convirtiéndonos en el mejor proveedor de servicios, junto con siempre estar en el campo de la innovación. Específicamente por siempre estar en el campo de la innovación es que hemos tomado compañías como Element Biosciences y otras que le vamos a presentar para siempre poder ofrecer el mejor resultado para ustedes con las mejores tecnologías que existen ahora mismo en el mercado. Para esto entonces les quiero hablar sobre cómo nosotros podemos dar una solución integral para la área de genética o realmente nuevamente cualquier área de biología molecular. Nosotros tenemos kits especializados para lo que sería la extracción de ADN, ARN, proteínas de diferentes matrices, desde las más complejas como heces, suelos, aguas, hasta matrices más universales como digamos sangre o plasma. Esto lo podemos hacer a través de varias compañías, pero principalmente desde la compañía Saimo, la cual fue una compañía fundada por investigadores para investigadores, el cual ofrece una alta variedad de kits de extracción. Después también tenemos las compañías Agilent y Bioptic para todo lo que es la cuantificación y cuantificación del ARN, ADN y proteínas, lo cual es un paso importante para cualquier determinación de cuál será la mejor material para poder hacer una secuenciación, es decir, poder ver la cantidad de su ADN y la calidad de este. También, como vieron, tenemos compañías de marca mundial o de renombre para lo que es la preparación de librerías. Tenemos Sophia Genetics para todo lo que es la parte clínica. Aquí ya sé que hay varios usuarios que conocen Sophia Genetics. Con esto podemos ofrecer todo lo que son soluciones para exomas clínicos, exomas completos, mieloide, linfoma, leucemia linfocítica crónica, aguda, todo con fusiones, sin fusiones y ya con una solución bioinformática incluida. También tenemos Agilent para los cuales serían otros paneles clínicos y Twist Bioscience que como vieron tienen una complementariedad con la tecnología de Element para lo que sería el Trinity y también tienen la capacidad de hacer cualquier otro panel custom para su interés en la secuenciación. Y por último tenemos las plataformas en sí. Tenemos tanto las plataformas AVITI de Element para lo que sería la secuenciación de lecturas cortas o la secuenciación multi-ómica, como los que serían la semilla PacBio para lo que sería la secuenciación de lecturas largas. En esto también poseemos lo que serían las soluciones bioinformáticas completas, nuevamente con Sophia DDM, los cuales pueden utilizar para poder hacer todo lo que es detección de variantes, detección de deleciones, lo que sería CNVs, toda la que es la interpretación con la facilidad de utilizar inteligencia artificial para ayudarlos a llegar a sus resultados y por último con la capacidad de hacer un reporte incluido tanto en inglés como español y de igual manera todo esto es compatible con lo que serían nuestras otras compañías. También el flujo de Sophia es compatible con el de Element, es decir, nuestros reactivos de ULP para Universal Library Prep de Sophia son compatibles con la química de Element y la misma data que nos da los equipos de secuenciación como el FastQ se puede fácilmente implementar a lo que sería el pipeline bioinformático de Sophia DDM para usted poder ver todo lo que sean las variantes que encuentran en sus muestras. Lo mismo es con la compañía Agilent, la cual es la preparación de librería de estos son compatibles con las mismas químicas de Element y también Element es compatible con los equipos y la bioinformática del mismo Agilent. Mucho gusto.